this is like Groundhog's Day, so thank you. We're going to start. I'm going to be talking to you about um, maintenance therapy, and I'll just put a little plug in for that. That is actually City of Hope. So if you don't recognize it or haven't been there, this is our new Orange County center in Irvine, and that's where Dr. Lee, who's going to be speaking later, and Dr. Chang are also located. And we're incredibly proud of our facility, and it is, as some of you have already visited, can attest, it's really um, quite a beautiful facility, as beautiful as a hospital could be, I would say. So I'm going to talk about maintenance and why do we do it? Why is it important? And what are we changing in maintenance? So you've heard a lot about this concept of minimal residual disease this morning. We talked about, and how do we check for it? And I recognize people are at different levels in terms of their knowledge about it. But really just to sort of codify it, our approach to myeloma has really changed as we've had better and better drugs. And what I mean by that is this concept of the iceberg. That's really, we talk about a lot in myeloma. So previously, we would be happy if patients got a complete remission. And so you think about that, that means that basically with the top of above the water, we don't see any more myeloma. And that's because if you look at this definition of complete remission in myeloma, it means you need to have less than 5% plasma cells in your bone marrow and no detectable M protein in your blood or urine. But think about that you can still have a lot of myeloma in your body. And that's the idea. There can be myeloma lurking under the surface. And in fact, there often was in those patients who we thought were in complete remission. As we got better and better drugs, we were able to chip away more and more at that myeloma that was lurking under the surface till we get to this really tiny amount that we can detect now by these really sensitive tests, and that's minimal residual disease. And you heard Dr. Patel say 10 to the minus 5, so 1 in 100,000 myeloma cells we can find, 10 to the minus 6, 1 in 1,000,000 cells we can find of myeloma now. So the question is, why is that important? And it's because this idea that bit of minimal residual disease can still take root and regrow and cause relapse of the myeloma. Now, it's a lot more complicated than that, though, because I'll give you an example. I saw a patient who had a transplant over a decade ago, is not on any treatment. Out of curiosity, we did a marrow, I think about a year ago, and she has minimal residual disease positive, but she hasn't relapsed in a decade. And so we think it's not just having minimal residual disease. We're learning more and more that it's also the immune system, or what we call the immune microenvironment, that also plays a role in terms of myeloma regrowing or coming back. So it's a complex thing. And so so this ideas will evolve as we get better treatments for myeloma, and certainly immune therapies are a huge part of that. Now, what do we use for maintenance right now? So the biggest tried and true drug we use is Revlimid or lenalidomide. And I've listed here, and you don't need to memorize them, all the studies, but the important things are, when you look at those studies, take home a couple things. Number one, those studies compare the use of Revlimid to placebo after stem cell transplant. And what they showed is if you give Revlimid versus just observation, you can almost double the time that people stay in remission. We then combined a couple of those trials together, and we did what we call a meta-analysis of over 1,000 patients, and found that if you used Revlimid maintenance after a transplant, you could actually make people live longer. And that sort of is the ultimate gold standard for us as myeloma doctors. Can you improve survival? And indeed, you can. So much so my colleague, Dr. Shaw, who I encourage you, follow her on Twitter. She tweets really only at the big meetings, but she's very witty and summarizes all the abstracts in very catchy ways. made the analogy that Revlimid is like the Tom Cruise of myeloma. It's been around forever, but it's still going strong. And so then we did another study, and I was the chair of this. This was a national trial. It's called the stamina trial, and that might be because of the stamina required to get through this, because one of the arms you can see was 2 transplants followed by lenalidomide maintenance. This trial was asked the question, what's the best thing to do after your first transplant to stay in remission? But we then looked later at this trial also for a couple other things. One of the things we did was look at, does it matter how long you stay on Revlimid? And so initially when we did this trial, we only planned to do it for 3 years of Revlimid. And that, quite frankly, I wish I could say that was science. It wasn't science. It was just practical. That's how much Celgene would pay to support for the 3 years of Revlimid. So that's initially how we started, because these trials often happen in parallel. So we didn't have all the other data yet about Revlimid maintenance. But then all the other data came out. Let me go back. And what we found was, in fact, staying on Revlimid maintenance beyond 3 years helped keep people in remission. So this sort of came out, and then we have all those other trials to build upon it. The idea of staying on Revlimid maintenance kind of indefinitely really became sort of the gold standard in the United States. Now, the French, on the other hand, still very differently, not surprisingly, use a fixed duration of Revlimid. They use 1 year in their study. Sometimes they use 2 in some of their other studies. And they did that because of this concern of second cancers. And so you can see that there is a higher risk of getting a second cancer if you're on Revlimid maintenance compared to doing nothing. But some of that becomes the devil you know, because the use of maintenance, especially with Revlimid, it reduces your risk of getting myeloma by over 50%. So it's a balancing act. The one good thing we learned is the risk of second cancer doesn't seem to increase the longer you stay on Revlimid. So that really then sort of gets us more interested in other research questions, part of which we're doing right now at City of Hope, of looking back and saying, can we predict who is at more risk of getting second cancers? Is it something in someone's bone marrow that already predisposes them to them? Is there some mutations that they have, for example, that they may be more likely to get this? And maybe those patients, we would in the future maybe not use Revlimid, think about something else. So this is, again, how the field evolves to more personalized medicine. So we're doing this balancing act. We're looking at, we want things that keep people in remission longer. We want things that make people live longer. We want things that are safe. So we also think about side effects, right? So we think Revlimid is great because it's a pill, but those of you on it know it's not that easy to take. Fatigue, diarrhea, skin rash, dry eyes, cost, certainly big issue. And this idea you heard from Dr. Chari this morning, I mean, everyone wants to be off of therapy at some point. So, are there patients who really don't need to be on it indefinitely? So trying to figure that out is important as we move forward. You heard this morning about risk stratification. You heard Dr. Chari mention the Forte trial. So we love catchy names for our studies. So this is an Italian study. And it's probably not important to sort of key in on the initial part. The initial part was trying to answer the question, do you need a transplant or can you just use fairly intensive chemotherapy? In this case, carfilzomib, lenalidomide, and dexamethasone. But looking at the second part of this, study, what did I just do here? Study right here, it looked at maintenance and it looked at Revlimid, standard maintenance, or it looked at a stronger maintenance, adding carfilzomib to the Revlimid. And I didn't put the results in here, but basically it showed, especially for high-risk disease, really augmenting the maintenance with carfilzomib seemed to help. So patients with 17p deletion, for example, patients who had that 414 chromosome abnormality, Patients who had 1Q abnormalities up to a certain point also seem to benefit. So it suggests the discussion we heard this morning. So for high-risk patients, we definitely do maintenance and often do even stronger maintenance than just Revlimid alone. Now, the next question, though, is of course, then if you're going to do these stronger maintenance strategies, can you use other tests to figure out when can you stop them? So this is a Polish study. And what this study did was give patients carfilzomib and Revlimid. It's important to note they only did it and then looked at patients who had standard risk disease, so not high risk patients. They gave them 6 cycles, and then they did that MRD test to see if they found any myeloma left. And if they didn't, they de-escalated therapy and just kept people on Revlimid alone. Now, I don't love this study, to be honest, but it is what it is. I use it more to illustrate a couple points. So what this study showed is if you did carfilzomib and Revlimid, you stayed in remission longer than just doing Revlimid alone, but you didn't live any longer. And this is the big question. What is the price you paid for staying in remission versus just waiting, and if the myeloma started to get worse, then adding another drug? So the price you paid is, first of all, a lot more visits to your doctor, 112 more infusions and visits. So think about that in terms of time and cost as well. Over $500,000 extra cost. So, these are the things you weigh and physicians weigh as you think about a risk benefit analysis, picking the best maintenance option for each patient. And that's why, again, it's not a one size fits all. And then you heard Dr. Chari this morning mention the MASTER study, and this is a study that's still ongoing. So, the conclusions may change or evolve as time goes on, but it does a couple things. It takes, first of all, probably the most intense regimen we have right now. So the quadruplet regimen, but and using daratumumab, carfilzomib, lenalidomide, and dexamethasone. So that's a pretty intense regimen. It is pretty immunosuppressive as well. Doing a transplant and then doing more of that regimen after the transplant. You can see here 8 cycles of that, looking at MRD at multiple time points as well. When you look at MRD, you also need to look at what test did they use. So here you can see they used 10 to the minus 5. So that's important versus 10 to the minus 6, just in terms of how sensitive your test was. And what they found is this idea of risk again. So they de-escalated therapy if patients became MRD negative. They then allowed patients to go on Revlimid alone. But they found that even in the high risk, ultra high risk patients, so those patients who had 2 or more of these high risk chromosome features, even those who got extremely deep responses, if they stopped or de-escalated therapy, they had a higher risk of disease recurrence. So it suggests for ultra high risk patients, you really need to keep the sort of foot on the accelerator and keep the intensity of therapy going. But for patients who had much less risk, you can stop therapy after a period of time if you get deep sustained responses. So this is how our thinking's starting to evolve in terms of maintenance therapy for patients. And then this is the trial that I'm leading right now. This is a now international study because we're doing it with Canada as well. It's over 1,000 patients. It's called the Dramatic Study. And we're really excited about this study because it's going to answer 2 questions. First of all, it's going to answer the question, is adding daratumumab to Revlimid a better maintenance option for patients? So can you get more patients into remission, stay in remission? And the ultimate endpoint of this study is really what we all want to know. Can it help patients live longer? But the other exciting thing this trial is going to do is say, can you stop therapy? So if patients after 2 years are MRD negative, down to 1 to the 10 to the minus 6, so 1 in a million, they get assigned to either continuing therapy or stopping therapy. So it will answer in a truly randomized, unbiased fashion if you get a prolonged, deep response to your treatment, can you stop your treatment? So, you know, so the good news is this trial has already accrued over 900 patients. It's accruing quicker than we even projected, which really speaks to, I think, the need for this study. The other good news is, as you heard this morning, patients with myeloma are living longer and longer. So that means that this overall survival endpoint is going to take years to really find out. And probably Dr. Patel is going to be the only one left sitting on the stage at that point. But you know, I think it is very important. We're of course going to look at other earlier time points to try and get a clue to that as well, but not wait till the ultimate endpoint. But, you know, I think that shows you why maintenance studies are very, very hard to do. They cost a ton of money because you need so many patients and you need to follow patients for years. So following patients for up to a decade to be able to get these answers. And then this is the future. So you heard about bispecific antibodies, these new drugs. You heard that teclistamab just got approved. We're incredibly excited about bispecific antibodies. So of course, in myeloma, the minute you get a drug approved in relapse disease, you want to say, can I use it somewhere else? So this is a study that's going to open using teclistamab plus Revlimid, comparing to Revlimid alone after transplant. And it's a fixed duration, 2 years in both arms, to see if that can improve and keep people in remission. So this trial's going to recently just opened. Again, big study. So it's going to take a while to get an answer. But again, it just shows you where the field is heading. So the other future direction is CAR T. Of course, everyone says, can I get a CAR T cell? So there is a trial, and we're literally having the site visit for it in the next couple weeks at City of Hope for patients after an autologous transplant and after 6 months of Revlimid. If they haven't achieved a deep response, they're eligible to get CAR T cell consolidation. So that's a trial. It's a national study. I talked to you about the Majestic study, the teclistamab study, and then I talked to you about really MRD. And I do see that in the future, that is really going to be a big part of our decisions and deciding when are we going to stop maintenance therapy. So let me conclude by what we want. We all obviously want to be stronger. We want to be faster. We'd all love to be Usain Bolt, but we have to acknowledge myeloma is a heterogeneous disease. We have to acknowledge as treating doctors and researchers, we have no agreement. What's the ultimate endpoint? We all know we want people to live longer, but are we willing to wait that many years to get that answer from a study? Probably not. Right now, what we know is there's certain patients we need to go stronger. Patients who have high-risk myeloma, for example, and ultra-high-risk myeloma, we really need to continue intensive, prolonged therapy. But there are other patients who have relatively standard risk disease who probably we can start backing down and taking the foot off the accelerator. And we're waiting for the results of the dramatic study to really hopefully prove what we've already sort of a priori thought in our minds. And then lastly, I do want to acknowledge all our myeloma group, which is growing and growing and adding in Orange County as well. I want to just put a plug in that we've opened our research operations in Orange County, and we've been very excited. The dramatic study is open down there. And we're going to start one of our first bispecific trials down there as well. So we're really excited to have our Lennar facility accessible to patients. So thank you. And speaking of Lennar, I am excited that we're going to have our next speaker, Dr. Lee, and I'll let Greg introduce him.